I'd like to introduce Julian Cardini. He is a PhD student at the Research Group for Analytical Food Chemistry, and he will be talking about the occurrence and bio-accessibility of essential and potential toxic trace elements in plant-based foods. Thanks very much, Julian.
Thank you very much for the introduction. Thank you. Okay, so I'm going to present to you my PhD project that is about the occurrence and bio-accessibility of essential and potentially toxic trace elements. But first of all, what even are toxic elements and what are non-essential elements? The essential elements are elements that are needed for the human, for normal functioning. Think of iron, for example. We need it in our heme protein because for the transport of oxygen through our body, and therefore it is essential. Then, of course, on the other hand, we have the toxic elements, and as the name suggests, they're toxic. And prime example for this one is, as your parents always told you not to play with the thermometer because it has mercury in it. And but now the question is, how do we actually absorb all these elements? And this most of the time happens through the gastrointestinal tract. And here we can talk about the bio-accessibility. And the bio-accessibility tells us about the proportion of a compound or an element that through the gastrointestinal tract gets ready for absorption from the body. And here we have four major factors that play a role in this process. That are first of all the food matrix, so how difficult or how deep it is in the cells, for example, to extract it. Then we have the nutrient-nutrient interactions. Think of the iron tablets when you have anemia. They oftentimes have vitamin C in it, so that the body can absorb it more easily. Then we have the processing, like heating it and cooking it, that changes the matrix. And then, of course, individual factors that come from genetics, because everyone is different. Then let's talk of my main interest. My main interest is cadmium, and cadmium is a highly toxic, heavy element. And the problem is that it's practically present in everywhere in the environment because of pollution, because of mining, of smelting, and but also there are natural occurrences where the cadmium concentration is higher in the soil. And of course, cadmium has its good side as well, since it can be used in batteries, pigments, and coatings. So, but what am I actually doing? I'm trying to develop a workflow for the speciation, so the identification of compounds that bind to cadmium. In this case, I can show you here a protein, but cadmium can attached to many different compounds that vary really from proteins to peptides to small acids. And the problem is the extraction of those acids or those compounds is really difficult because of their inherent instability. And for the analysis itself, I'm using size exclusion chromatography, so chromatography that is that separates the compounds based on size, and reverse phase chromatography, where the separation is based on hydrophobicity of the compounds. For measuring those compounds, I'm using inductive coupled plasma mass spectrometry for the elements, so for the metals, and high resolution mass spectrometry for the proteins, for example. Afterwards, we're going to compare the cadmium fingerprint of different whole foods. I always like to use the fingerprint analogy because if you think about it, every sample, every food has a different amount of every compound, and this builds a fingerprint that is unique to every food, you could say. And what's going to happen is that we are going to try to measure as many samples as possible, and then using multivariate analysis, we can see, okay, where is the difference between those foods? Where is the cadmium bound? Lastly, we're also going to assess the bioaccessibility of those cadmium species. As said earlier, these they're very unstable, and during the digestion, what happens is that the pH changes, and due to this, also the species of the cadmium species changes. and could therefore change also the bioaccessibility of those compounds. And yeah, thank you for your attention, and we hope that we can make this food safer.